Hello everyone, today we will talk about a new topic called Central Dogma of Molecular Biology. This is an extremely important topic in molecular biology. This concept of central dogma was proposed by Francis Crick in 1958. Francis Crick is an important scientist who discovered the structure of DNA along with Watson. And this concept, Francis Crick told us that the flow of genetic information occurs from DNA to RNA and to protein in sequence. So it is the sequential flow of genetic information which occurs from DNA to RNA and to protein in sequence. This phenomenon of central dogma occurs with the help of three processes or steps. First is DNA replication, next is transcription and translation. DNA replication is a process in which a copy of DNA is made with the help of DNA polymerase enzyme. So this process preserves genetic information which is required by each and every new cell forms because they need a copy of DNA. So for that, DNA replication is an extremely important process. Next is transcription. Transcription is a process where an RNA copy is made from DNA by using RNA polymerase enzyme. So here the flow of information occur from DNA to RNA. Next is translation. In translation, protein is formed from RNA. The information present at RNA and ribosomes helps in this process. So here the flow of information occur from RNA to protein. But this is the usual flow of information from DNA to RNA to protein, but there is an exception to this normal rules. In retroviruses, for example, HIV, where the genetic material is an RNA molecule, so they use the information present in RNA to make a copy of DNA. So the flow of information occur from RNA to DNA. So by the special mechanism known as reverse transcription, and the enzyme they use in this process is known as reverse transcriptase. So retroviruses uses a special mechanism known as reverse transcription. In that process they made DNA from RNA. So this is an exception to the usual flow of genetic information which occur by the process of central dogma, in which the flow of information occur from DNA to RNA and to protein. Now we will discuss the diagrammatic view of central dogma. This is the simple version. You can see here a DNA molecule. From this DNA, by the process of DNA replication, a copy of DNA will be formed. And the DNA molecule, by the process of transcription, results into an mRNA molecule. And from mRNA, by the process of translation, a protein will be formed. And this is the detailed version of central dogma. Here a double-stranded DNA is given. The one strand is given in black color, the other strand is given in blue color. The black color strand is known as coding strand, and the blue color strand is known as a template strand. From the template strand, an mRNA molecule will be formed. This is known as transcription process, and the sequences of mRNA will be the same as the coding strand. But in mRNA, thymine will be replaced by the uracil. Also, the information present at the mRNA is in the form of triplet known as codon. You can see here, this is one codon, this is another codon, this is another codon, and this is another codon. This is the simplest view, and we only show a few codons. The number of codons depends on the size and the type of protein formed. So this codon, each codon code for a specific amino acid. So AUG, it code for methionine amino acid. Also it can act as a start codon, and it can start the process of translation. So this codon has a dual function. This codon CAC, it code for histidine, and this codon GGG, it code for glycine. The last codon we shown as UAG, it acts as a stop codon, and it terminates the process of translation. We know that at least 20 amino acids are required to make a specific protein. So this is all about the central dogma of molecular biology, which states that the sequential flow of information occur from DNA to mRNA and from mRNA to protein.